Okay, yeah, thanks a lot, Carsten, for the introduction. It's a great pleasure to present some of the work that we have been doing at Genetech, and thanks a lot for taking the time today to listen in. And ideally, I would like to make this interactive. Obviously, we can't make this an interactive session, but definitely feel free to pepper me with questions at the end of the talk. So I'm happy to take any question. So coming to the first slide, so it's as you all know, drug discovery is a very risky endeavor that is prone to high attrition rates. And it's also a well-established fact that much of the attrition is due to toxicity. What I'm showing you here on this slide is a study that was published about 2 years ago in Nature Reviews Drug Discovery. It's an analysis of the attrition that happened in 4 companies. So it's an analysis done at people from AstraZeneca, Eli Lilly, GSK, and Pfizer. And they look at the entire portfolio from candidate nomination all the way to late-stage clinical phases. And as you can see here on the left-hand side here, the indicated in blue are all the compounds that are trit in these individual phases. You see massive attrition from candidate nomination all the ways to late-stage clinical phases. Then on the right-hand side, you see the reasons for the attrition. So you see that number one is non-clinical toxicity. So this basically refers to most of the compound that are trit in the stage of candidate nomination. And then also another point is clinical safety. So these are compounds that are trit during phase 1, phase 2, and phase 3. So definitely toxicity is a major reason for attrition. And seeing these statistics obviously begs the question, can we do better than that? And especially for me, since I'm a medicinal chemist, the question is, what can I do as a medicinal chemist to remedy the situation? So I believe that as a medicinal chemist, I can do a lot here. I mean, typically for drug discovery programs, we have counter screens, so we can build in selectivity against an off-target, and so we can do this with structure-based design and all the basically techniques that we have at our disposal. Or we also know that certain properties predispose compounds for off-target effects. So typically compounds that are very lipophilic, or especially when they're associated with high basicity, such compound we know they are prone to high off-target effects. And then if we don't know if the toxicity is caused by the target of interest or by another target, we can find out through tool compounds that we provide. We can try to resolve the question: is the toxicity caused by on or off-target on-target effects? So definitely, there's a large role that medicinal chemists can play here. And what I'm going to present to you in the next 40 minutes or so is a case study where you can see basically all these elements. You can that are described here. So you can see how we strive for high target selectivity. We strive for good properties to basically evade the propensity of off-target effects. And then also finally, we provide tool molecules to help resolving the question of on and off-target toxicity. And the case study that I'm going to present to you today is about a kinase inhibitor program, PAK1. So real brief. So what is PAK1? So PAK1 is a kinase, also called P21 activated kinase. It's a serine threonine kinase in the so-called ST20 kinase family. It's a downstream substrate of the G protein called RAC CDC42 and has important roles in governing proliferation, survival, and motility. The PAKs are subdivided in 2 groups, group 1 and group 2. Group 1 comprising PAK 1, 2, and 3, and group 2 comprising PAK 4, 5, and 6. And within these groups, there's very high homology in the kinase domain. So essentially, there's almost very little difference between the kinase domains of within these 2 groups. So the PAK that we are predominantly interested in is PAK1. And why is that? PAK1 is amplified in nearly 8% of all breast cancer. That means it exists in gene copy numbers of greater 5. So that's typically when we call it a meaningful amplification. Especially very high in the poor prognosis luminal B subtype. And luminal B subtype is a subtype of estrogen receptor positive breast cancer, and it's known to be a very aggressive subtype. And also in our labs, we found out that cell lines that harbor PAK1 amplification are particularly sensitive to PAK1 knockdown. So all these data together, the clinical data and also the data from our lab, built a very strong case to launch a drug discovery program on PAK1, and that's what we did. So I'm showing you on the, oops, my slide is stuck. Sorry, technical issues. I can't forward my slide. Try that again.
Yeah, maybe if you, oh yeah, there you go.
Okay, it works now. Sorry about that. So I'm showing you here basically the road to a lead series on PAK1. So we started like as we very typically for a drug discovery program with a high throughput screen. But what we encountered very early on were low hit rates, and and all the hits were mostly non-selective. And what we gathered from our own observation, also from observation that were reported in the literature, that PAK1 is very malleable. So the kinase binding site and ATP binding site is very malleable. We believe that this compromises the ligandability of PAK1. But nevertheless, we were able to identify our starting point. So this is shown on the left-hand side. So this compound here, this was our HTS hit set, but it's a very promiscuous compound. It's hitting a lot of different off-target kinases, and it was very, very challenging to build in reasonable selectivity against PAK1. But we were successful with that, and also this compound here is an advanced lead compound that we identified. So it's more potent against PAK1 than the starting point, also has nice selectivity against PAK4. And this compound has excellent kinase selectivity, and the way we achieve that is by building in this pyrrolidone ring here, which interacts with a pre-DFG moiety called threonine 406, and it builds a very specific hydrogen bond to that threonine 406, and it gives this compound very nice selectivity. We reported this data in ACS MedChem 2015. However, the selectivity came at a price, and that price was poor permeability and lack of cellular potency. So if you look at this compound, it's a very polar compound. It's hydrogen bond donors, hydrogen bond acceptors, very high polar surface area, and we believe that this is the reason why this compound wasn't very permeable. And then we tried, obviously, to to build back the permeability, but whenever we tried that, we lost the kinase selectivity. So we weren't really able to balance both properties, the selectivity, together with the permeability. And at the end, we had to make the tough decision to abandon that series. But very fortunate for us, we got the opportunity to in-license another series, and the series was offered to us by a company called Afraxis. And Afraxis had a very nice starting point shown here. So this is the main compound that we in-licensed, called Frax 1036. So that's a very PAK1 potent compound, nice selectivity against PAK4, good cellular potency, and also excellent kinase selectivity, and kinase selectivity shown here. So very similar actually to the aminopyrazole series shown above. However, obviously this wasn't a candidate compound yet. It had some liabilities still, and these liabilities were low ligand lipophilic efficiency, which is basically a measure of how efficiently lipophilicity is used to impart potency. So this compound was relatively lipophilic still, potency not quite in line with that. So it had low LLE, and also it had ion channel off-target activities. So obviously, to address low ligand lipophilic efficiency, there are two ways to do that. You can concentrate on lipophilicity, or you can concentrate on potency, or you can do both. Initially, we tried to find ways to further increase the potency of the compound. What we focused on initially was the tail on the left-hand side. And what we found out, we made about 100 analogs. So this was very easy to do that with library synthesis. We made about 100 analogs, but we realized very quickly that we cannot increase the potency through this effort. What we found out through multiple X-ray structures that this basic amine chain actually doesn't really have any specific interaction with any residues of the protein. So it's basically merely a solubilizer and not really driving potency. We also then asked the question, is there anything that we can learn from our previous series, the aminopyrazole series, any pieces that we can incorporate into the Afraxis series to build potency? And very interestingly, the aminopyrazole series also has a basic amino tail, but that amino tail is actually directed towards a different region in the protein. It's actually directed toward the ribose pocket and has some very specific interaction with the aspartate 393 residue in the ribose pocket. And we knew from our work on the aminopyrazoles here is that this amino group is really a critical potency driver. So then we asked the question, obviously, is there a way that we, so here you see an overlay of the Afrexis series, the aminopyrazole series, and we asked the question, is there an opportunity to reach that aspartate 393 from the Afrexis scaffold, and would that give us basically the same potency that it does for the aminopyrazole series? And the answer is, we were very happy to see that the answer is yes. So we basically changed the vector. We appended this amino group here to this vector here, and made this compound 3586. And this compound is very potent. So it was really a major gain in potency, and a major gain in ligand lipophilic efficiency. You see here the original starting point, ligand lipophilic efficiency 2.8. Here, ligand lipophilic efficiency 4.2. So typically for drugs, we're striving for values like 5 and higher, so to give you a rough baseline here. And we also obtained an X-ray structure for this compound, and as we had hoped for or planned for, we really see indeed this hydrogen bond with the aspartate 393, as in the case of the aminopyrazole series. However, this compound still wasn't a candidate. It had an excellent pharmacology profile, but it had very poor permeability. We attributed that poor permeability to high pKa. So this amine here has a pKa of roughly 10, so very basic. So we believe that this compound is too polar and too basic. That gives it the poor permeability and accordingly very poor exposure when we dosed it in mouse pharmacokinetic studies. And so we spend a lot of time then reducing the basicity of this amine, but still maintaining the good properties, still maintaining reasonable solubility of this compound. This was really a balance act, so it was very challenging to find such a compound, but we were able to do so. And to do this, we had to resort to a very unusual group. So this is this aminodioxane group. So I think the gut feel reaction for medicinal chemists is to cringe whenever you see a residue, whenever you see a group like this, because you would intuitively expect that such a group is very unstable or very acid unstable. But it turns out that this group is actually quite stable. And the reason is because the amino group actually protects, the presence of the amino group protects the acetal from hydrolysis. So it's a very, very stable group. We did a lot of stability studies really to confirm that this group is perfectly stable. I can actually highly recommend when you have similar problems to try out this group as a medicinal chemist. But then the question obviously was, does it help us with permeability? And the answer is yes, it did help us. You see here the permeability value of the parent compound 3586, and then with this change here with the lower pKa group is a pKa of 7.7, we had improved permeabilities. It's not particularly high, I would say, but high enough. And we also removed the efflux. But then the big question, obviously, what does it do when we do such a compound in mouse pharmacokinetic studies? We can see here, this is the original compound, very low oral bioavailability. And for compound 555, we were very pleased to see that this compound had very high oral bioavailability, 80%. And then also even more pleased when we dosed this compound in higher species, in the cynomolgus monkey, likewise very high oral bioavailability. So with this, we had a very, very good compound in hand. But another question also that I wanted to address is, so I mentioned that the original lead compound that we in-licensed from Afrexis was very promiscuous. So the question is, how did compound G555 fare in terms of promiscuity? So you see here on the left-hand side, the secondary pharmacology panel is a panel from so-called SERA panel that we're using, which has ion channel and various ion channels and receptors. We see here a compound 1036, the original starting point is a very promiscuous compound, and then compound G555 is a much cleaner compound, and we attribute that to both lower log P, basically reduce it from 4.9 to 2.9, and a lower pKa. And then also on the right-hand side, you can see the kinase panel profile. So this compound was G555 was very clean compound, actually more selective than the original starting point. So with that, we had a very good starting point in hand that we could proceed with to in vivo studies. So before I start with the in vivo studies, I would like to hand it over to Carsten to do a quick poll. So Carsten.
Yeah, of course, we would like to know a little bit more about our audience, and we prepared a quick poll. There'll be one more poll as we go along, but this poll here is really a little bit for you guys to describe yourself, what you are. Are you a medicinal chemist, computational chemist, crystallographer, molecular biologist, or other? So that could be a toxicologist, physicist, physician, or any other discipline. I'll give you a little more time to make your selection. We seem to have a clear winner here. Okay, I will close the poll in 5, 4, 3, 2, 1. And it seems like we have mostly medicinal chemists in the audience, followed by the computational chemists. There are a few molecular biologists here, and then 17% describe themselves as other. And with that, I will hand back to Joachim.
Okay, thanks, Carsten. So I mentioned in the previous slide that we were able to identify a really good tool compound. So this was really the first time in the PAK-1 program that we had a good tool compound in hand that we could proceed to, that we had sufficient selectivity, sufficiently good pharmacokinetic properties that we could proceed to in vivo studies with. And what we started out with is a mouse PK/PD study. We did this in the H292 non-small cell lung cancer cell line. And I'm showing you here the outcome of the PK/PD studies. So in red, you see the exposure doses 10, 20, and 30 milligrams per kilogram. So you see a nice dose proportional exposure. And then in terms of PD marker, our PD marker of choice was PMEC S298, which is a very specific residue for phosphorylation that is mediated by PAK-1. You see also that we imparting very nice dose proportional suppression of the PMEC signal. So far so good. So what we saw there was very promising. Then we proceeded to mouse efficacy studies, and you see immediately that the responses were actually quite disappointing here. So what we did here, we looked for tumor growth inhibition upon oral dosing of compound G555. The time span is 15 days of the study, and in green you see the vehicle, so the mice that were not treated with compound, and the other lines here are mice that were treated with compounds. You can see immediately that the effects are very weak. Actually, the highest effect that we were able to achieve was 25 milligram per kilogram BID, was only 60% tumor growth inhibition, so quite disappointing. And this turned out to be the maximally tolerated dose for this compound. And whenever we dosed higher than that, we saw acute lethality. So the mice were actually dying very quickly after being dosed with the compound. So I give you a little bit more granularity on the toxicity that we saw. So when we dosed, I mentioned we dosed higher than 25 milligram per kilogram, we saw the death. So for example, the dose group 40 or 50 milligram per kilogram led to death 2 to 4 hours after the first dose. So what were the observations? The observations were the mice were hyperactive, cold to touch, the blood was dark and hard to draw, and the blood flow of these animals was very slow. The blood flow of these animals was very slow. Then also we knew that this compound is not getting into the brain, so we did multiple studies to show that the brain plasma ratio is very low. So the effect that we saw was unlikely to be driven by a CNS driven effect. I show you here the concentration plots. This basically shows concentration. This is the phosphomec IC50, 70 nanomolar. Then in green lines, you see Cmax values from PK studies that were associated with doses that were tolerated. And then you see in red here Cmax values that were associated with doses that led to death of the animals in the study. As you can see here, the window between the PMEC IC50 and the Cmax that's associated with death of the animals is very, very small. So there's really not much of a therapeutic window here. Coming back to the observations that we made in this study, so the observations clearly suggested cardiovascular toxicity. When you make this observation of slow blood flow and blood is dark, hard to draw, that is often suggests cardiovascular, not always, but often suggests cardiovascular toxicity. And based on these findings, then we proceeded to mechanistic safety studies. And I'm showing those to you on the next slide. So one very I would say popular study to do if you suspect cardiovascular toxicities is the so-called Langendorf heart, which is also called the hanging heart. It's an ex vivo study. Basically, a heart from an animal, for example, from a rat, is taken. It's paced like basically so that it behaves like a normal, like an in vivo heart, and this heart is perfused with compound and at multiple doses. So when we did this study with the Langendorf heart, what we saw vasoconstriction and impaired relaxation of the left ventricle. So left ventricle is that compartment in the heart that is basically pumping oxygen-rich blood through the aorta into the systemic circulation. We saw that the left ventricle essentially is no longer doing its job. And then we also, so this was an ex vivo experiment. We also did an in vivo experiment, and that is mouse echocardiography. So we're basically creating with using ultrasound, and we're creating with ultrasound images of the heart. And so what we saw, basically I can summarize that very quickly. So what we saw when we dosed these animals with the compound, very similar findings as what we did in the Langendorf heart. Basically, major finding was that the left ventricle is not doing its job, and this severely decreased cardiac output and performance. So definitely we had a problem with this compound, and our first gut feel response to this was there's got to be some off-target effect, because the effect kicks in so quickly, and it has very much kind of the look of a ion channel, and that's what we first suspected. So what do you do when you expect an off-target effect? Then you try to modify your lead compound or your lead series with the goal to dial out that presumed off-target activity and maintain your on-target activity at the same time. So that's what we did. And so I'm showing you here basically the fruits of about like half year of medicinal chemistry work. So this is our starting point, compound G555. So what we tried to do, there's still a relatively lipophilic head group in compound G555, and we tried to replace it with a more polar group. And we were able to accomplish that with this pyrimidone group here. And another effort that we took is try to completely rid the compound of an amino tail. And we were able to do that, and we put in this methoxy ethyl tail here. And we are very pleased to say that we had a compound with quite a different look and also different physical-chemical properties. You can see here the starting point here, G555 at a log D of 2.5, CPK of 7.7. Here with compound 9479, we have a compound that has even lower log D, 1.7, and essentially a neutral compound, so no longer any basicity present. And so typically when we have compounds like that, we're definitely taking a strong effort to reduce the propensity for off-target effects. And that's exactly what we saw. So you can see in the serial panel here that this compound 9479 is very clean, so barely hits any off-targets. And likewise, also in an vitrogen kinase panel, this compound is very selective. But the big question, obviously, is this compound free of toxicity? And the answer was very disappointingly no. This compound showed the same toxicity as compound G555 did, and with a very similar therapeutic window, and also very similar clinical, basically similar animal observations. So this compound is causing dark blood, and the blood is flowing slowly. So very similar phenotype as with what we observed with compound G555. And that was the first time when we really suspected, oh, this could be some on-target toxicity. But obviously these observations that were made still on compounds from the same series, and I think it's much easier to convince oneself if these observations are being made with compounds from entirely different chemical series. Unfortunately, at that point, we didn't really have any series that had sufficient oral exposure. So what we had to do, we had to resort to a different way of dosing the compound. We used interperitoneal dosing. So the two different compound series that we dosed are shown here. So one is the compound from Pfizer, PD3250901. This is a compound that actually proceeded into the clinic as a PAK4 inhibitor. It's actually really a pan-PAK inhibitor. And we saw with this compound, as you can see here, likewise toxicity when we dosed it interperitoneally with a very similar window. And likewise also a compound from another series that we worked on, the aminopyridine series. Likewise, we saw toxic effects in the very similar window as observed with other compounds. And all the observations that we made when we hit the toxic doses were very similar as with compound from the Afrasiab series. So clearly now we have 3 chemical series with all these compounds we seeing toxicity. Then just to convince you further, I want to show you basically a Venn diagram and showing you the kinase activities of those 3 compounds. This is the Pfizer compound shown on the upper left here, and compound G555 on the upper right, and compound G9479 at the bottom. And you can see here really the only overlapping kinase activities of these 3 compounds are PAK 1, 2, and 3. Then we did a similar analysis for a Sera panel, and I'm not showing the slides here, but the outcome of this exercise was that there was really no overlapping activity in the Sera panel of these 3 compounds. So clearly suggesting that PAK 1, 2, or 3 are the culprits for causing the toxicity. And I also want to show you that we looked at compound of various range of cellular potencies. So all the way from very potent compound shown here, this compound 9791, to compounds that are less potent. So this compound even caused death of the animals in a low dose PK study. This compound, you had to dose it very high to induce death of the animals. But as you can see here, the correlation is actually really, really good. So across this entire range, we have good correlations, basically a log range of 2. And again, these are compounds from different chemical series with different potencies. So clearly suggesting that what we're dealing here is with this on-target toxicity. And basically finally to put the nail in the coffin, I'm also showing you here an active-inactive pair. So this is this compound 1608. So when we move this alkyl group here to the para position, we are dialing out completely PAK 1 activity. And when we dose such a compound with no PAK 1 activity, we weren't able to find any toxic dose for this compound. So this compound is very safe and doesn't cause any toxicity. So clearly another piece of evidence that we're dealing with on-target toxicity. So with that, I would like to hand it over one more time to Carsten for, I think, the second and the last poll.
Yes, we have one more poll here for you, and we would be interested in your opinion. If you could do magic, what would be sensible in a piece of software for modeling medicinal chemistry applications and so forth? Would it be minimizing the small molecule and the protein, do flexible docking, meaning having a flexible backbone on the protein, do selectivity profiling, which means you can calculate the affinity in several proteins at the same time, do on-the-fly patent checking, meaning if you design a compound, you can check on the fly whether this compound has already been patented, or any other suggestions which we would like to hear from you at support@biosolvit.com. I'll give you a little more time to decide. This is obviously something that's a little more, invokes a little more thinking than what am I? Am I a medicinal chemist or a biologist? I'll give you a little more time and I will close the poll in 5, 4, 3, 2, 1. Okay, interesting. So 40% decided on do a selectivity profiling. I can tell you that this will likely be coming at some point in our CSAR software. So you'll be able to do an affinity calculation in multiple proteins. You can already load multiple proteins, but so far you can select only one binding site at a time. The next one in is do flexible docking, and then do on-the-fly patent checking with 16%. Okay, with that, I would like to hand back to Joachim.
Okay, thanks, Carsten. So I showed you in the previous slides basically all the measures that we took to demonstrate that what we're dealing here here with is on-target toxicity, and I'm just summarizing on the next slide basically all the things that we did. So first of all, we could demonstrate that the toxicity is present with chemically diverse inhibitors. So we could show that 3 chemotypes, the Aphraxis, the aminopyridine, and the Pfizer series all caused acute lethality in the mice with very similar observations made. There were also no overlapping off-target effects when we looked at CREB and kinase panels. Then we could show that the toxicity is absent with an inactive analog. So when we had an inactive pair, only the active analog was toxic. And finally, we could also demonstrate, or thirdly, we could also demonstrate that there's a correlation between the target potency and the toxicity. So the lethality correlated well with the cellular potency. We did this analysis with that compound, and it was across a 300-fold potency range. And one thing that I hadn't really talked about is the target actually expressed in the tissue with the toxicity. And the answer is for PAK1 and PAK2. PAK1 and PAK2 are widely expressed, and including in the cardiovascular system. But what about the other PAK1 group 1 PAK group member, which is PAK3? It turns out that PAK3 is actually predominantly expressed in the brain. And I could already show you earlier that our compound is not getting into the brain. So really what we're dealing with here is either PAK1 and PAK2 or both causing that toxicity. So then very importantly, is the toxicity consistent with the phenotype of a knockout mouse? So before we started the program, actually, we had PAK1 knockout information. And from that, we gleaned that a PAK1 knockout is actually viable and fertile. And that was for us one important reason to consider a drug discovery program on PAK1. So how about PAK2? So for PAK2, we actually did not have, when we started this program, embryonic knockout data. But there was a group in the Fox Chase Cancer Center led by Jonathan Chernoff that actually did some studies on PAK2 knockouts. And he found out that PAK2 knockouts are embryonically lethal. And the phenotype is vasculature development. But obviously, for embryonic lethality, that's not necessarily a death sentence for programs. We know many of the kinases, for example, the MAP kinase or EGFR. So many kinases are associated with embryonic lethality. Yet it doesn't mean that such targets are not viable as drug discovery targets. However, it's more concerning when an adult conditional knockout leads to lethality, and that's exactly what's the case. So, the group of Jonathan Chernoff later also did adult knockout studies, and likewise he observed lethality here. But nevertheless, even an adult conditional knockout is not exactly the same as when we inhibit a compound with an inhibitor. It's not the same as completely removing the target through a knockout. So what would really be the best model is a kinase-step knock-in, but such a kinase-step knock-in was actually never generated. So it's not really completely. You cannot really completely compare a knockout with the condition of inhibiting a target. But nevertheless, coming back to the question, is the toxicity consistent with the phenotype of a knockout mouse? And definitely, based on what was learned about PAK2 knockouts, it is definitely reasonable to assume that PAK2 is involved in the toxicity. So here's our hypothesis. So our hypothesis is that PAK2 is the driver of toxicity, and it's not clear is it PAK2 on its own, or is it combination with PAK1? So that question is not entirely answered yet. But obviously, our target of interest is PAK1, and so since we believe that PAK2 is the predominant driver of the toxicity, what about a compound, a PAK2 sparing PAK1 inhibitor? Would that have a path forward? And before we ask that question, we have to ask the question: is it actually possible to identify such a compound? And I'm showing you here an image of the PAK1 ATP binding site. And here in magenta, you see basically the difference between PAK1 and PAK2 and the ATP binding site. So overall, it's the ATP binding sites are very, very similar. And there are really only two differences. This is here in the back pocket here, this methionine isoleucine difference, and here this tyrosine phenylalanine difference. So these are obviously very similar residues. And also the side chain here, the phenylalanine or tyrosine, are pointing away from the ATP binding pocket. So very, very difficult to leverage these residues for selectivity. And that's exactly what we saw. So here you see all 2,400 compounds that we made in the program, and you see really not a hint of any compound that has a PAK1 versus PAK2 selectivity. However, we were very fortunate that actually another company also worked on PAK, and they described a PAK1 selective compound. And this compound is actually an allosteric compound, so-called type 3 kinase inhibitor that's not interacting with the hinge, but predominantly sitting in the back pocket. So they described this compound at a fall ACS meeting in 2013. That obviously really, that was when our program was still going on, definitely sparked our interest, and we synthesized the compound, and we could confirm indeed that this compound is a very selective PAK1 inhibitor. So here the PAK1 Ki, and here the PAK2 Ki. So in our hands it had 60-fold selectivity, also very selective over PAK4, and overall the kinase selectivity of this compound is very high. So very, very strong tool compound. However, the problem with this compound was, and that was totally in line with what Novartis described, this compound has no cellular activity. So in our phospho-MEK assay, the phospho-MEK IC50 was 1 micromolar. Also, we tested the compound in various proliferation assay, and it was consistently inactive. I should then also mention that Novartis did a very interesting experiment. They combined this compound with PAK2 knockdown, and when they combined this compound with PAK2 knockdown, then the cellular potency came back. That basically suggests that PAK1 and PAK2 have redundant functions, and you really need to inhibit both to get the desired cellular effects. And there was, so what Novartis described, we also tried to corroborate Novartis findings, and we completely could basically completely made the same findings that Novartis made. And we also couldn't identify cancer cell lines that were dependent on and would express only PAK1. So really, there's a high redundancy between PAK1 and 2, and you really need to inhibit both to have the desired pharmacological effect. And coming to the conclusions, so simultaneous inhibition of PAK1 and PAK2 provides cellular efficacy, but it is associated with severe cardiovascular toxicity. Selective PAK1 inhibition is insufficient to promote a cellular efficacy, but it's presumably safe based on the PAK1 knockout experiments that were described. But overall, there's no path forward for a PAK1 inhibitor program because you can't really evade the toxicity. And so this basically prompted us to abandon the program, and since we didn't really see any path forward to get out of the safety problem. So one important lessons learned that I wanted to highlight to you is definitely it's known that kinases can be implicated in cardiovascular functions, and there have been cardiovascular toxicities described for kinases, but the acuteness and the severity observed here for PAK1, PAK2 was very uncommon, and for the longest time we really didn't believe that a kinase can actually do that. And for me, that was a very important lesson that we always need to watch as drug discovery scientists our preoccupations. In that case, that preoccupation was acute cardiac toxicity needs to stem from an ion channel. So it took us actually really quite a while to convince ourselves that inhibition of a kinase can cause very acute toxicity. So with that, I come to my last slide. So the title of the presentation was Resolving the Question of On or Off-Target Toxicity. And I wanted to come back to what can we do to build evidence, just in general, for any drug discovery program. But first of all, I think what's very important, what I hopefully conveyed to you, is it is important to have a strong set of tool molecules. And these tool molecules need to be sufficiently selective. Then ideally should be structurally diverse, and ideally distinct series. And what is desirable also to have compounds of varying cell potencies. And then very powerful is the use of active-inactive pairs. And then also sometimes, as I showed to you, the compound from the Pfizer series or from our aminopyridine series, those compounds didn't have sufficient oral exposure, so we had to resort to intraperitoneal dosing to get sufficient in vivo exposure. So here the lessons is definitely consider enabling technologies if drug exposure is an issue. You don't always need really the ideal compound with ideal oral exposure. There are some enabling technologies, and intraperitoneal dosing is just one of them, but there are many other enabling technologies that can be used to give compound the sufficient exposure. Then, second, target expression knowledge is very important. So for the on-target toxicity, the target must be expressed in the tissue with toxicity. So for example, if you observe toxicity in the lung, but your target of interest is not expressed in the lung, then your target of interest cannot be the root cause for the toxicity. So it's very important to pay attention to where the target of interest is expressed and where the toxicity is observed. And then finally, genetic rodent models. Genetic rodent models obviously are very powerful. There are a lot of different flavors of genetic rodent models. There's the simplest one, embryonic knockout, and then there's the conditional knockout, where basically chemical triggers used to knock out a target. And then there's these more sophisticated genetic rodent models, embryonic function deficient knock-in, for example, the kinase dead knock-in, or the conditional function deficient knock-in. And so typically we think of the conditional function deficient knock-in to the condition that really resembles most, is closest to the condition of using a drug to inhibit a target, but it's also probably the most difficult one to use. But nevertheless, the use of genetic rodent models also has caveats, and it's really the concerted use of tool molecules, genetic rodent models, and the target expression knowledge that kind of in concert makes it very powerful to generate evidence for on-target toxicity. So with that, I'm at the end of my talk. I would like to thank the people in my team for staying dedicated to the very end. So obviously our mission as drug discovery scientists is to identify clinical candidates, which obviously did not happen in this program. But our mission is also to seek the truth and design make or break or go or no-go experiments whenever we have the opportunity to do so. As you could see, we did not shy away from such experiments, and that allowed us to come to a very firm conclusion, and still in the research phase of this program. And with that, I would like to thank you for staying to the end and listening to the seminar, and I would like to hand it back over to Carsten.
Yes, thank you, Joachim, very much for this interesting talk. That was really a great presentation. And while you were presenting, there were a few questions coming in. Are you available to answer a few of those?
Yeah, please.
Okay, so the first question was, what is the best way to predict off-target? And did you use any software to do that in your experiments there?
Yeah, I think there are a lot of publications on basically what predisposes compounds to off-target. I think there's this very kind of brush stroke way to predict off-target. Typically, compounds that are very lipophilic, compounds that are very basic, are prone to off-targets. There are multiple analyses have been published from people from Pfizer, AstraZeneca, multiple other companies that show that. That's basically, I think that's very general in drug discovery. That's something that we observe all over and every time in programs, that when we are in that space of high lipophilicity and high basicity, especially combined, then we tend to hit a lot of off targets. I mean, for example, HERC is a very prominent example when there's lipophilicity that is log D greater than 4, and it's paired with high basicity, then your likelihood of hitting the HERC channel is extremely high. There's a very nice study, I think, published by people from AstraZeneca. But these are more, I would say, empirical data that have been published. That's probably, I would say, the most generalizable guideline for us in drug discovery programs. But I think specifically to predict off targets, I think general off target, I think that's probably the best that we can do, because you're dealing obviously with a lot of different off targets, and you're staying away from high lipophilicity. I would say that's probably the best that we can do as a medicine chemist. It's not always simple to do that, because some targets really require lipophilicity, and then it's just an inherently higher risk.
Yeah. Okay, thank you for answering that. Then the next question is, could toxicity be related to residence time? If the inhibition is virtually irreversible, perhaps you need to change this to reduce toxicity?
Oh yeah, absolutely. I mean, residence time is a very big topic, and I think I didn't really talk in my presentation about residence time. And it's something, I guess, for off targets, it's an unknown what is the residence time for your off target, but we can measure it for our target of interest. And I would say for off target, since it's really an unknown, I'm not sure to what extent I can comment on that.
Okay, thank you.
It's probably the best I can answer.
Sure, no worries. Okay, another question was, I don't know the slide number, it's not given in the question, but what does FL Wuxi on the y-axis and NVS PAK1 mean?
Yeah, NVS PAK11 is just that's the name of the Novartis compound. The Novartis compound is actually in the chemicalprobes.org. It's a very nice chemical probe because it's such a selective PAK1 inhibitor, and that's the name that has been assigned in the chemical probes listing. This name was actually not mentioned in the Novartis publication, but I think later on it's basically the common name for this compound. And what was the other question for?
FL Wuxi, that was probably on the same slide. There, go back to the slide. On the y-axis, PAK2 FL Wuxi KI on the.
Oh, okay, so, yeah, I think our PAK2 assay was run at Wuxi. That's all. I probably forgot to delete that.
Okay, this is a question I think that's rather directed towards us here. How can biosolvate tools be leveraged to reveal selectivity changes, potency drivers, pockets, residues, and the presence of a neighboring pocket? Yeah, so this can be done in CSAR, not everything yet, but so with respect to the first thing here, selectivity changes, what you can do now is, of course, calculate the affinity in different pockets sequentially. You can already load several proteins at the same time. You can define a binding pocket for calculation one after the other, but you cannot calculate the binding affinity in two proteins at the same time. But there's a small workaround for that. Potency drivers, yes, you can already do that. If you have your compound in the pocket and you modify it, you can identify regions that are particularly adding value to the affinity. So these will be visualized by these large green spheres that are located on the atoms. So if you have a bunch of compounds and they all have the same large green sphere, we call them affinity corona, in the same pocket or area or next to the same residue, then you can, I guess, make the assumption that this would be a potency driver. And the presence of a neighboring pocket, we can calculate all potential pockets on the protein when we do the binding pocket selection. So if you don't have a molecule, or if you would like to find an allosteric pocket, for example, you can calculate all possible binding pockets and therefore will detect any pockets that are located next to the actual binding pocket or active site. Okay, then we have another question. When evaluating the cause of toxicity, what is the proposed sequence of events from PAK1 or 2 inhibition to cardiac dysfunction?
So what was the sequence? I guess as I showed in the talk, I mean, we had a very good tool compound that we thought was selective enough to dose it in vivo, and so we are always very careful and mindful if we have compound with low selectivity and we see toxicity. Obviously, you have to always be watchful and suspicious that off targets may cause toxicity. But we started out with a compound that was reasonably selective, and When we then saw the toxicity, I guess the first thing to do is to modify your compound substantially and watch what happens to your toxicity. I would also say another thing that could be tried when you see toxicity. Obviously, if you have a hypothesis on the nature of the toxicity, you can try to if there's a simpler way to find out what is the root cause of the toxicity, you can try to you can develop an in vitro assay or some kind of an assay that has more high throughput, and then you can use that particular assay to test new compounds in, which is obviously much better way to proceed with than putting a lot of compounds into in vivo studies and killing a lot of animals. So I think if you have an hypothesis on what the nature of the toxicity is, it's always good to basically work with your safety group and develop such assay if there's such an opportunity. I would say structural modifications, and then also I guess the point that I really didn't make, it is incredibly valuable to have compounds from different chemical series, because if you endorse compounds from a different chemical series and you see very similar toxicity with a similar phenotype, that's definitely highly suggestive of an on-target playing a role. So I would say I don't think there's a general recipe of how to go about once you make the first observation of toxicity, because it's highly dependent on the nature of the toxicity, on the kind of true compounds that you have. I think the way that I presented to you is one way of going about that in our particular case. Overall, I would say the use of full molecules, the use of genetic rodent knockout data, together with the target expression knowledge, those three things are kind of the generalizable items that I think that I think are very useful or important to get to the ground if the toxicity is on target or off target. But in what sequence to use those, it depends. But one case I really would want to make is the use of genetic rodent models is extremely important. And often I think we think of that, it's going to take a really long time, it's going to be really expensive. But if you think about how expensive it is to have a very large chemistry team working on the program, and I think the costs are actually relatively minor to actually early on and to prepare rodent genetic knockout models. Especially now we have very modern technologies like CRISPR technology, which actually allows to generate rodent knockout models much cheaper and faster. And so I think that should really be something that when you basically make a commitment to a target, should always be something that should be considered early on. Obviously, care needs to be taken. I think it's just, as I said, there are caveats, and the data coming from a genetic rodent genetic knockout model shouldn't be overinterpreted. It's just one piece in the puzzle, a very important piece. But I think it is. I would not, based on a knockout study, if there's lethality seen with the animals, it shouldn't be a reason to based just on those data to abandon a program. It's just one observation, and then you can develop hypotheses. You can use this information, but I think the way we use the data coming from such a study should be we should be mindful of how we use those data.